Okay, so this audio lecture is going to look at factors that affect microbial growth. So it's kind of two parts. There's the microbial growth, just a couple slides on that. And that comes from chapter 5 in our text, the food microbiology textbook. And then we'll look at factors that affect microbial growth. And that's going to come from chapter 6. There's also some figures that are from the general microbiology textbook that's available online. So the boundless version. So looking at microbial growth, microbes are going to grow by multiplying in number. And this happens when there's favorable environments. And so this can happen in food. And again, in a medical microbiology class, we think about this happening in a host. For bacteria, the process they undergo is binary fusion. So this is a replicate DNA, grow a septum, and separate and repeat the process. So you can see on the left figure how we have in A, the cell marked A, that's going to be an early stage of cell division. B is a little bit later. You can start to see the septum forming. And then C is where we actually have separation. There's still attachment that's happening. So they're right close together. So this kind of gives you this appearance of this streptobacillus, but those could further separate out, possibly. On the right-hand side is just showing kind of a classic drawing of binary fusion. Again, that happens in bacteria. And so as microbes grow and divide, they're going to have generation time, so the amount of time it takes to do that. If we look at an entire population, we look at the doubling time for the population, because we want to keep in mind that not all cells within the population, like if we look at a colony growing on a plate, they're not all going to be dividing at the same time. It's not like, whoop, they all split and start the process. They're going to have different times in which they're splitting. So again, it's just a doubling time is how long does it take the population to double in size. But if we were to look at the generation time, the figure below kind of shows you how you go from the 1 cell to 2, and then 2 to 4 to 8 to 16 to 32 to 64, and that's usually where I top out. But again, they keep doubling, and the generation time varies between organisms. So like E. coli has a 20-minute generation time if given, optimal conditions. Where soil bacteria tend to be slower. And again, all of this varies on the conditions that they are growing in. So obviously if we're depriving nutrients and some of those intrinsic factors, and we change some of those extrinsic factors that we'll talk about later on, like temperature, then this can change the generation or doubling time for an organism. So this is showing a growth curve of a bacterial culture, but the same process would happen if we had like a fermented food, or if we had a, if you're looking at medical microbiology, a host. And so the first phase is the lag phase. So this is where there's no increased number of cells, and the cells are acclimating to their environment. So if we just, inoculated or put our starter culture into a fermented food like cheese, there's going to be a little bit of time where the cells are acclimating. There's actually just a new paper out that looks at what's happening during log phase, and traditionally when we think about the lag phase, we think about it being kind of this like static, they're not doing anything, but it's actually a very dynamic point. And so I will post that article, the link to the article on our page, so that you can read it if you want to. There's a podcast that goes with it too, so it's actually a very interesting paper, and again, it kind of shifts how we think about the lag phase. What we traditionally think of as a very dynamic time for the bacteria is the log phase. This is also known as the exponential growth phase. This is when the bacteria is growing and dividing, and so we have biosynthesis, protein synthesis, new cell walls being built, and everything. So again, we think of this as being a very dynamic time for the cells because they're going and this is when they're doubling So we're going from 2 to 4 to 8 to 16 to 32, etc. So we get an increased number of bacteria cells. And then we'll enter into a stationary phase. This is where it kind of plateaus off. The rate of cell division and death is equal. At this point, the organisms have used up a lot of the nutrients that are in the material that they're growing on. So if we're talking about in the lab, we would have to change out the media. If we're talking about like a fermented food, we might have to provide a new food source. So this is where with your sourdough starter, you have to add more flour and water because again, after 24 hours, we've kind of reached the stationary phase. And if we don't do anything, we'll go into the death phase. By adding a new source, by adding the flour and the water, we enter back into the log phase. So we super concentrate our starters so that we have material that we can use to make sourdough. Now, as I mentioned, if we don't replenish this nutrients, if we don't change the media, they'll enter into death phase or decline phase, and this is where there's more dead cells. So the number of living cells are decreasing, and again, this is because they've utilized those intrinsic factors, so they can't support cell division. So next, looking at the factors that affect microbial growth, there's many ways in which we can either inhibit or help microbes as they're growing. And so there are going to be intrinsic factors and extrinsic factors that we'll look at. And so again, we want to keep in mind what type of microbe are we looking at? What is the function of the food? If we're talking about a fermented food, we want to make sure we provide a beneficial environment for the microbes to grow so that we end up getting that product of fermented food. If we're talking about food that shouldn't have the microbes in it, or we don't want pathogenic types of microbes that cause foodborne illness, then we need to think about how can we change either the intrinsic or extrinsic factors so that it inhibits microbial growth. So looking at intrinsic factors, this can range from nutrients, growth factors, inhibitors. Inhibitors act as antimicrobials, water activity, pH, and oxidation redox potential. For oxidation redox potential, we'll focus more on the oxygen requirements, but also within this looks at cellular respiration and the availability of oxygen. So they kind of overlap in that case. Looking at extrinsic factors, those are ones that are affected by how the food is stored or during the processing of the food that need to be controlled by the environment. And so temperature, relative humidity, and gaseous environments are all extrinsic factors that anyone in a processing facility or at a facility that is retail that's going to be selling food, and also us as consumers at the table need to be thinking about. So looking at nutrients, nutrients is all about making sure that we can support microbial growth, again, if that's what we want to do, so that the microbe can synthesize those cellular components and also have energy in order to support itself. So there are different necessary nutrients for this to happen. And again, where they're going to be getting this nutrients is through their environment, and in this case, it's going to be the food. The nutrients that a microbe would need are carbohydrates, proteins, lipids, minerals, and vitamins. So not so different from you. So if we think about any kind of cell, those are the building blocks. Those are the compounds that they would need in order to make those larger structures in order to support cell division. So looking at carbohydrates, all microorganisms that are normally found in food would be able to metabolize glucose, so they can be used that. But then they would also be able to utilize other carbohydrates. So there's a number of different monosaccharides, disaccharides, polysaccharides, that they would also be able to metabolize in additional, depending upon what enzymes they have and what pathways they can utilize. For proteins, there are a number of different protein sources that are found in food. So just as we as humans think about protein sources, microbes are also thinking about that. So foods that do have a protein source, some microbes would be able to utilize that. So the pathways in order to break down certain proteins vary. So they wouldn't, most microbes are able to have a preferred protein source. At the end, what they're going to end up doing is transporting the amino acids and small peptides into the cell. So it's not that they're transporting the whole proteins. They're actually going to do external digestion. So they're going to release enzymes, and those enzymes will break down the proteins in the food, and then they will absorb the individual amino acids or those small peptides through diffusion across the cell membrane. So again, just looking at some of these different food sources, albumins and eggs would be a protein source, globulins in milk, and then you have glutens in cereal, just in collagen and muscle. So you have lots of grains would also have protein. So lots of different, depending upon the food, would indicate which protein would be available. And then that would indicate which microorganisms could grow. Looking at lipids, examples of lipids would be free fatty acids, glycerides, phospholipids, waxes, and sterols. And so these are usually found at higher concentrations in animal products, but they also can be found in plant products, in nuts, and some of the oils isolated from seeds, and like avocados and olives actually have a very high lipid amount. And so this becomes really important when you're looking at prepared foods and what the lipid content is and how that then affects what microbes can grow. So looking at minerals and vitamins, there are several minerals and vitamins in which the organisms need to get up to consume, to take in through their environment. And most foods would have those in sufficient amounts. Some microbes can actually synthesize vitamins or for themselves. So many microorganisms can actually synthesize vitamin B. So they wouldn't rely upon that from their food source, but also foods would contain like vitamin B. Okay, and looking at some of the elements that they would need from their environment, phosphorus, calcium, magnesium, iron, sulfur, and like potassium would all be very important for them to consume for the microbes to consume in their diet. And again, that is coming from the food source. Another intrinsic factor is inhibitors. So inhibitors can either be natural or synthesized. And so this can be added. So if they're naturally occurring, they're already there. And if it's synthesized, they're going to be added into your food product. So secondary inhibitors are ones that are added in. Secondary inhibitors can be natural or synthesized. So just, I don't know if that confuses it more. So some food has natural inhibitors in it. So if we think about like plant matters, plants are already going to be producing inhibitors to inhibit microbial growth because their environments, just like us, we produce inhibitors naturally. Plants are going to do that too, right? But we can also add in additional inhibitors. So if we are adding in the inhibitors, those would be considered secondary inhibitors. And again, the producer could opt to use natural inhibitors, so they could add lemon juice in, or they could have isolated gluten from milk that they could add in to inhibit the growth of microbes in their food. Or they could synthesize these inhibitors in the lab and then purchase those chemicals and add them in. So definitely consumers are kind of pushing towards a more natural approach, and so you do see some of that, lemon juice added in, opposed to putting in like citric acid, because again, there's this huge fear in society of like chemicals. All chemicals are bad. Well, a lot of these chemicals originate from natural sources. It's just you only can grow so many lemon trees, right? So if you can synthesize citric acid in the lab, that means you don't have to have like an orchard with lemon trees. But the whole purpose of the inhibitors are to prevent or reduce the growth or even kill the microorganism. The next intrinsic factor is looking at water activity. So AW, or water activity, is the measure of availability of water for biological functions. And for microbes, this means can they grow? So microbes are going to require free water in order to grow. So if that is present in the food, it's going to be more likely that microbes can grow on it. So you can see in this table how, if we think about some of those dry, shelf-stable products like cereals and crackers or your sugar, they have a relatively low water activity. So there's not a lot of water available, so that actually inhibits microbes from growing in it. And this is why you could leave your cereal out, and you don't have to like refrigerate it to prevent microbial growth. And things like noodles or honey, again, they're going to have relatively a higher one, but still a relatively low. If we look on the higher end, this is where we have like fresh meats, fruits, vegetables, milk, eggs, and just think if you left those on the counter with that high water activity, there's going to be free water in order for microbes to grow and end up spoiling your food. So one of the things we can do is take those products and dehydrate them, or so we don't even have to dehydrate them all the way, but we can add either inhibitors to them, or we could, make, if we have fresh fruits, we could take those and make those into jams. That decreases the water concentration in it, so it makes it safer. But then we also go through the process of canning it. So we put it under pressure so that the microbes are less likely to grow. So again, we can take additional steps in order to take something that has a high water concentration to decrease that. The next intrinsic factor is looking at pH. Most microbes, bacteria fall in the neutrophil category, so they like a pH that is around 7. And so this works out because in us as humans, most of our tissues are at the pH of 7.2. So the same kind of microbes that we would find living in us tend to be neutrophils. There are bacteria that like a more acidic environment. They're acidophiles. And then we also have ones that like a more basic environment. Those are alkophiles. So each microbe is going to have an optimal growth pH range. And so again, if we're looking to stop the growth of a microbe by making a product slightly more acidic, by adding some acid into it, that can shift the pH so it no longer favors microbial growth. So this basically just says everything that I had just said. So we're going to move on and talk about oxygen. So oxygen concentration is really important for a microbe. And so obviously in a food product, depending upon how much surface area it has, would then indicate how much oxygen is available. We can also go through processes where we remove the oxygen, so we can vacuum seal products to create a different condition to be less favorable to microbial growth. But we definitely have microbes that can kind of grow on all spectrums. So if you look at tube A, you can see what's being shown by those big red spheres. They'd actually be like little. You would notice in a test tube that you'd have this like clouding up at the top. That would be an obligate aerobic bacteria. Aerobic needs oxygen. where the other extreme in 2b is an obligate anaerobic. And so you can see where the bacteria are down at the bottom. So the test tube actually creates this concentration gradient. So up at the top is going to be high concentration. At the bottom is going to be low concentration. So again, those are our two extremes. We can also have facultative anaerobes. So they're anaerobic, so they don't require oxygen, but they still can live in an oxygen-rich environment. So you can see how they're kind of really found throughout the test tube. They're similar to the aerotolerant anaerobes. So you can see these are also found throughout the test tube. So again, the terms of aerobic and anaerobic environments are utilized in talking about oxygen. Aerobic being where we have oxygen present, anaerobic, oxygen's not present. When we're talking about an environment, just be aware that those terms are also used in cellular respiration. In a bacteria that undergoes anaerobic metabolism, that deals with what is the terminal electron acceptor. And so just because a bacteria undergoes anaerobic respiration doesn't mean it's an anaerobe. So it might be able to live, just like you saw on the last slide, it might be able to live in an area that does have high oxygen concentration. Okay, and so just like these little niches within our body with different oxygen concentration are found, we would also find those in food. And so this just kind of defines those terms that were in that previous figure. So just like there is an optimal pH, there's going to be an optimal oxygen concentration for cells, for microbes, and so they're going to grow fastest in whatever their optimal oxygen concentration. There are several enzymes that allow for microbes to deal with oxygen. Peroxidase, superoxide dismutase, or SOD, and catalase are some of the main enzymes that help to detoxify reactive oxygen species. So when you have oxygen present, it ends up breaking down and it forms these reactive oxygen species. And so these enzymes are able to bind those reactive oxygen species so they don't damage the cell. So for your aerobic bacteria that like high concentrations of oxygens, they would be more likely to have all three of these enzymes. And if we look at anaerobes, obligate anaerobes, they may not have any of these enzymes. Okay. So then looking at extrinsic factors, the main one we're going to focus on is looking at temperature. So again, extrinsic factors are going to be ones that we as producers or consumers or in retail can control a little or more. So we can control the temperature, we can control relative humidity in a climate-controlled setting, and we can also control gaseous environments. So this would relate to that oxygen and other gases that would be present. So looking at temperature, microbes can have a preferred temperature range, just like they do with acid and oxygen and our other factors. If we're looking at medical microbiology, they would fall into the mesophile category, which you see in that yellow, and that is because that correlates with body temperature. If we're looking at environmental microbes, we could have thermophiles. Those are commonly soil bacteria because soil tends to get nice and warm, and it retains some of that heat. If we were looking at like thermal vents and lava fields, we would have the hyperthermophiles. And then if we were looking, up at Antarctica or looking at icebergs and snowbanks, we would have the psychophiles. So those like really cold temperatures. So again, mesophiles would be medical microbiology. Because with food we tend to be looking at food sources that are either coming from, an animal source or looking at plants, products that have been grown. Mesophiles and our thermophiles would be the two categories where our food microbes fall into. When we're looking at fermented foods, those tend to be thermophiles, so they tend to like a slightly warmer temperature. And we can use this to our advantage because we can change the temperature, right? So we can change the thermostat, or we can utilize devices like a refrigerator or a stove in order to change the temperature so that we can inhibit growth of these thermophiles and also the mesophiles. So again, we can expose the food to high heat. So one, this would denature some of those proteins so they wouldn't be available for the microbes to grow, but it also would be outside of the range of some of those mesophiles that potentially could be foodborne illness, cause foodborne illness. So heating up to 65 degrees Celsius or even up to 100 degrees Celsius would allow us to remove a lot of those potential foodborne pathogens. If we were going to do a long-term storage, because obviously we can't, heat the food for a long time, this is where looking at the other extreme and actually refrigerating, so 5 degrees Celsius or even freezing, which can be down to negative 20 degrees Celsius, would give us more long-term storage. Now there are some stable foods and that can be kept at room temperature, and sometimes that's because we've actually altered the water concentration that they're able to be shelf stable. And then there's also ready-to-eat foods. So ready-to-eat foods should be kept at a warm temperature so that so again, we're moving out of that mesophile category so that it's no longer favorable for them, and that food should be consumed shortly thereafter, within several hours. So looking at these the intrinsic and extrinsic factors will affect the microbes that are present, so how many microbes are present. And so there's a process called quorum sensing. And so quorum sensing plays a role in how the bacteria is actually going to respond to its environment. And again, this is another topic that's looked a little bit more in medical microbiology, but obviously plays a role in food microbiology because quorum sensing could be a positive in the bacteria now is going to metabolize and ferment the food, and you got your fermented food, but it also can be in a negative sense in that you have enough bacteria present, they quorum sense, and now they become pathogenic and they cause foodborne illness. So quorum sensing is a way for bacteria to communicate with each other. They produce small chemicals, small molecules that they have receptors for, and they have different chemical signals they can use. So they have chemical signals that are kind of general signals. I liken this to like a smile. It doesn't matter, what language you speak, a smile is understood. So bacteria have that universal language, but then they also have a special language that only within their species knows. And so this would be our individual language or dialect that we have. So we even have like regional dialects, right? So there's just like, E. coli would have its own dialect, or, Salmonella would have its own dialect. So they have their own chemical signal that they only have receptors for. So what they can do is actually count how many cells are out there in general, and then how many of those cells are the same species. And if there's enough of them, then they can change gene expression. They could produce toxins. They could produce digestive enzymes. And again, this changes their behavior and how they interact with their environment, whether that environment is food or whether that environment is their host. So this figure just looks at quorum sensing. So quorum sensing plays a role in whether a bacteria is going to produce spores, whether they form a biofilm, whether there's some kind of survival factor that is initiated, and that could be survival from like an immune response, or survival from some kind of inhibitor that's been added into the food. And again, it's all kind of feeds back into these virulence genes being upregulated and expressed. And so again, this huge area of research in medical microbiology, but definitely looks like it plays a role in food microbiology and an area of research within the food microbiology community. So this concludes the audio lecture, looking at microbial growth and also factors that affect it.